I got interrupted there. I'm sorry, but we're just going to keep going with this concept. So I think we left off right here. So these two we said were purebred. This was purebred dominant. This one was purebred recessive. And I think I also added that the synonym of purebred was homozygous. Same. Synonym of purebred, homozygous. So this one's homozygous dominant. This one is homozygous recessive. Now, the one in the middle is not homozygous or purebred. It's called a hybrid. Now, I know we're used to the term hybrid referring to cars that use both an electric motor and a gasoline motor, but in this case, hybrid means they have both the dominant allele and the recessive allele present in their genotype. So this particular plant is a hybrid. Now, it's a hybrid that is always going to show the dominant allele. It just carries this one around. Now, it could pass that trait on to an offspring, but it does not show that trait because the dominant one shows, the dominant one is present. Okay, you don't have to say hybrid dominant because everybody knows that a hybrid plant is going to show the dominant allele even though the recessive allele is also present. But it does have a synonym, and that synonym is heterozygous. Okay? So a hybrid plant or hybrid organism would be heterozygous. Hetero means both for that particular gene. Okay, now there's one other term to know, to understand before we jump into then how to use these in Punnett squares, which is going to be next. Okay, so that's the term phenotype. The word phenotype comes from the root word comes from the same root word as phenomenon, okay, or phenomenal. It's simply the gene that shows. So it's the allele that expresses itself, the allele that shows up in an organism. And we already know that. We've already talked about that. We just haven't used the word phenotype yet. So in these two, the allele that shows up, the allele that expresses itself, is going to be the purple allele, the dominant allele. So these then have the phenotype purple. This one is going to be the only one of the three that has the phenotype white. Remember, the lowercase p stands for white. Okay? So if you're going to use this word phenotype, you would say, well, the phenotype of this flower is purple. You can say the phenotype even if you don't know what the genotype is. But you can also say the phenotype when you know the genotype because you know the rules. If the dominant one is present, it's going to show, whether it's purebred or hybrid. If all you've got are the recessive alleles, that's the one that's going to show, and that's going to be the phenotype. Okay, so you've got all of these expressions. I want to then introduce one other idea. And this is the idea of the Punnett square. Punnett square. Actually invented by a lesser-known geneticist named Punnett. But this is simply a way for us to be able to figure out what the genotypes and phenotypes of offspring are going to be if we know the genotypes of the parents. So I'm going to just give you two examples on the board, and then we'll have to get together on a call of some kind to be able to do a few more examples after you've practiced to make sure you understand how these work. So let's say that we have a Punnett square, and this is our Punnett square. It just looks like a 4-square board, like a window. Okay, that's the Punnett square. Now, it doesn't really matter which side of the square you put which parent's alleles on. or which parent's genotype on. But for our purposes, we'll just say this one's going to be dad and this one's going to be mom, okay? So let's say that we have the same situation as we had a moment ago with flower color. And we have purple that is dominant. So let's say that we've got a dad that is homozygous dominant for the trait flower color. He's got 2 purple alleles in his genotype. Let's say that we've got a mom, or the female genotype here. Let's say that that mom is going to be white. Now, if her phenotype is white, we know that her genotype has got to be homozygous recessive. And we use the 2 lowercase p's because we already learned that the recessive allele is expressed as the lowercase letter of whatever the dominant allele was. We also know for this particular trait that that recessive allele is going to be white. Okay, so here's mom, here's dad. Now what the Punnett square is going to help us do is figure out what are the chances that the offspring are going to be one color or another for this particular trait of flower color. What are the chances? Now we've got 4 squares. Each of those squares represents a 25% chance, or 1/4 chance. Let's work the Punnett square, and then we'll talk about the chances in a minute. So we work this just like you would work a multiplication table. You combine this with this. By convention, sort of the rules are that you always write the uppercase letter first if it's present, but otherwise it doesn't really matter which one you write. So here in this square, we'd combine the capital P with the lowercase p. That means that this square represents a 25% chance that you're going to have an offspring with that genotype. Of course, the phenotype here would be capital P, purple. This one, voila, it's going to be the same thing. The uppercase P combines with the lowercase p, and we get a heterozygous or hybrid in that square as well. Got another 25% chance. What's going to happen here? Well, you guessed it. You've got the capital P combining with the lowercase p. Got another 25% chance. And then in this 4th square, you've got that uppercase combining with that lowercase, and you've got another hybrid or heterozygous 25% chance. Now what all of this is telling us is that 100% of the offspring are going to have the genotype, capital P, lowercase p. Even though dad was purebred for purple, and mom was purebred for white, or dad was homozygous purple, mom was homozygous white, all of the offspring are going to be purebred or heterozygous for those traits. But their phenotype this was the genotype. Their phenotype is going to be uppercase P, purple. They're all purple. They're all hybrid. They all show up as purple, but they all carry this other allele within their genetic makeup, and they could pass it on to the next generation. So let's look at that next generation. Let's say over here, we've got a different scenario. Here's a new Punnett square. And in this Punnett square, we have a hybrid plant and another hybrid plant for this particular genetic trait, flower color. Okay, So we do the same thing. I'm going to not draw the arrows this time because they get a little bit confusing on the Punnett square, and I don't want you to either. I just did that so you can kind of see how I was doing the Punnett square. I'm going to do it over here, just kind of showing you. So you combine the capital P with the uppercase P, just like if you were doing a multiplication table. And in this square, we've got a dominant, so you have a homozygous dominant, purebred dominant. In this square, you have the uppercase from here and the lowercase from here. We wrote the uppercase first because that's just the rule of using Punnett squares. So uppercase, lowercase. This square, we're going to have the uppercase from up there and the lowercase from over here. Uppercase, lowercase. So we have another heterozygous or hybrid chance there. And here we have the two lower cases. Lowercase, lowercase. So this Punnett squares yields really different results when we crossed two hybrid plants. So the way this would be done is you take the pollen from one plant and you put it on the flower of another plant. And in this case, both of the plants would be hybrid plants for that flower color purple. And what is this telling us? Well, this square represents 1/4 chance. So there's a 25% chance that you're going to get the genotype capital P, or homozygous purebred dominant. That's this one. There's also a 25% chance you're going to get the genotype lowercase, or homozygous recessive, purebred recessive, okay? And then these two are each 25%, so we add them together. 25% plus 25% equals 50%. So there's a 50% chance that you're going to get the genotype capital P, lowercase p, okay? Those are the percentages of genotypes. Now let's think phenotypes. What's really going to show up? Well, let's see. This one's going to be purple, this one's going to be purple, and this one's going to be purple. So, from a phenotype perspective, I've got 75% that are going to show that purple allele, and I've only got a 25% chance that we're going to show the recessive allele, which is white. So even though both of the parent plants would have had purple flowers, that one was purple hybrid, that one was purple hybrid, even though the parent plants were purple flowers, if I combine the pollen onto the flower and they have seeds and I plant those seeds in the next generation, I've got a 25% chance that the seeds that I plant are going to be white. Or, another way to think about it, if I plant 100 seeds, I probably am going to get 25 of them that are going to be white, and the other 75 will be purple. So that's the way that you use a Punnett square. Now what I'm going to do for the next video is I'm going to give you some additional examples of how to use the Punnett square in the same way. What I did just now was Punnett squares using what we sometimes call classical genetics, and that's the idea that there's a dominant and a recessive allele for any given trait. You'll also see that coming up there's going to be some practice questions for you to be able to do some Punnett squares on your own. Okay, so this is part 2 of the video. Hope that you gained some knowledge, and please send me an email or a text or give me a call if you have any questions. Good luck.